Hi, we're going to be going through the process of how ribose and nucleotides are made and how they come together to make ATP molecules, along with how they are used for DNA replication. I will also be going over the ATP production and regeneration of ATP synthesis along with the rest of the processes that are involved. The main focus of this will be on the ATP synthase, and ATP synthase is an enzyme that creates energy storage. It's an energy storage molecule, and it is most commonly used in energy currency in all life forms. It forms adenine diphosphate and inorganic phosphate. ATP synthase is localized in the inner membrane where it is catalyzed, and it involves it involves the flux of protons that is generated in the electron transport chain, and where they go from chemically positive to chemically negative, where there's a difference between the two of them. For ATP production, most of the ATP that is in the cells is produced by the enzyme ATP synthase, which will convert the ADP and phosphate into ATP, which is localized in the membrane of the structures and specifically in mitochondria. It can also be found in the chloroplast of plant cells. However, what we're going over for is specifically produced in the body. ATP is created through a complex enzyme-driven process where there are a few different ways. There are a few different ways that this will work out. Glycolysis is when glucose is broken down into two different subunits called two different subunits of pyruvate, which has been taught to us throughout this course and through this project. This process will allow us to create 2 units of ATP per molecule and of glucose that occurs in the cytoplasm. For the relationship of ATP and DNA, ATP is used to create the DNA and RNA in the body. Adenine is a base of DNA and RNA, and you can add a sugar molecule and 3 phosphates to it to create one of the bases for building DNA or RNA, which is adenine. I said that. 2 ATP molecules are used in the preparation of the RNA primers, and ATP is also used as an energy source for some of the enzymes that are used in this process. Specifically, ATP is used at the replication fork. Nucleotides that will be incorporated with the DNA chain that is growing continuously is selected by the base pairing and is for the template DNA. By the template DNA. DNA replication is a basic process that occurs in the cell, and every time that a cell divides, the daughter cells that form will also have the exact same genetic DNA, or the exact same genetic material or DNA, because of this process. Every DNA strand can be used as a template strand for DNA replication. DNA replication is important due to the fact that it occurs in all organisms and allows for the cells of the body to replicate themselves and create new cells and allow for the body to age and grow throughout one's life. It is also the way that all biological life can pass along their genes and create offspring. With the aid of glycolysis, the enzyme catalyzed reactions that occur in this process will ultimately aid in the replication of DNA and RNA, which will allow for the body to function correctly and the cells to be functioning properly together to allow for them to work in a way that allows for the essential metabolic processes to occur for the body. So essentially, the DNA process needs to occur for the body to be able to function properly and in the way that it needs to be. Overall, this is a breakdown of the glucose molecules for the production of energy for the body and pyruvic acid. Glycolysis allows for there to be a metabolic pathway that converts glucose to create free energy that is released while making the pyruvate, and is also used to form higher energy molecules that we know as ATP and NADH. As this process occurs, there is a sequence of 10 enzyme catalyzed reactions that essentially result in the DNA replication process. The main 4 steps that occur during this process, there are 4 main steps that occur. The first, the helicases unwind in the parent DNA strand, and then a singular strand of binding proteins help to stabilize the unwound parent DNA strand and continue with the leading strand being continuously replicated while the lagging strand is replicated in segments known as Okazaki fragments, and they'll be connected later using a binding enzyme. After the RNA primer is replicated by the DNA ligase will join the Okazaki fragments from the lagging strand to the growing strand. These processes come together to create the replication of DNA using ATP and glycolysis through pyruvate. By using these processes, it allows for the body to maintain its own form of homeostasis and allow for it to function properly. And this is my whole poster, and I hope that you have enjoyed my presentation for our Advanced Concepts 8. I really enjoyed this class. Thank you so much.